To what extent is your health destiny predetermined? Is it nature or nurture? What you eat and how you live that determines your health history, or the genes you're born with? My guest in this podcast is a specialist in nutrigenomics. Now, that is the field of rich interactions between your genes and your environment. Emma Beswick is not only a nutrigenomic expert, but also a graduate from the Institute for Optimum Nutrition. So she knows her stuff about nutrition too, and is very much hands-on running the best gene testing company in this field called LifecodeGX. So Emma, welcome to my podcast.
Thank you, Patrick, and I'm really delighted to be here. Thank you for the wonderful introduction too.
A pleasure. And have you looked at, hundreds or thousands of people's gene analyses? What's your kind of experience in that side?
Yeah, it's many thousands, probably over 10,000, maybe even 20,000. So I've been doing this for 8 years, living and breathing it and being quite obsessed, really.
Well, it's fantastic to actually have that hands-on experience. And I want to really, give the idiot's guide to genes, because there are so many words. We hear polymorphism, SNPs, genome, phenome. The whole field is rich with a new vocabulary, which to Joe blogs is extremely confusing. And yeah. I'd like to wind about when was the human genome finally unraveled fully?
Oh, fully. It was late last century. It's really evolved as a science quite rapidly. During the 1900s, most of the work was done, and it's just picked up at a rate of knots and gathered pace. And there were all these imaginings that it would be the solution to all of health's challenges. And it has delivered lots, and it still is delivering lots, but we are far from knowing all the answers.
And I remember when that sort of big reveal was, how many genes have we got and all the rest, which is really not long ago. I mean, we're talking, 20 or so years ago. The concept was, in a sense, that we'd have lots of genes because we are the most superior of all species. And if I remember rightly, it turned out that rice had more than us. And I believe there was a water flea that had sort of 31,000 genes, while we had 23,000 genes. Wasn't it something like that?
Yeah. So more is not necessarily better. And I think strawberries are another life form that have more genes than us too. So yeah, it's not about quantity for sure.
And let's start exactly at the beginning. What is a gene?
So a gene is a collection of letters. Some people might describe it as a word. So it's a string of letters, but there are only 4 possible letters to play with. So it's not a very complicated alphabet, but those letters can be arranged in lots of different ways. And the smallest grouping is of 3 letters. So those 3 letters make up something called a codon, and that codes for an amino acid. And then those codons are chained together, and they together code for a protein. So it's a bit like the gene is a sentence made up of words that are themselves made up of letters. So just these building blocks that are all chained together to create an instruction that indeed can be read.
And those letters are literally, when we see the chromosome as that spirally thing that you often see in various advertisements, and it's got the the rungs of the ladder across that spirally chromosome.
The
And
helix.
the helix, yes, the double helix. And I don't know, do you know the story about how Linus Pauling didn't get his third Nobel Prize?
No.
Well, in essence, having won 2 unshared Nobel Prizes and being his second, which was for Nobel Peace Prize, for his campaigning and taking over from Einstein to campaign against nuclear bombs. As a consequence, he was grounded, labeled a communist, passport confiscated. And in his spare time, he was working on the structure of DNA. And he really wanted to go to Oxford University to meet Madeleine, the French scientist who was doing some experiments. It's always like this in science. You've got the theory on one side and the experiments on the other. But he wasn't allowed out of the country. So he guessed a triple helix. And Watson and Crick just got the Nobel Prize. It's a double helix. And they said, if Linus Pauling had been able to get out of the country and get the data we had, he would have beaten us to it.
Wow. It's a bit like the space race or something, isn't it?
It's just like the space race. And then when you talk about these letters and 3, make a codon and so on. So is it fair to say that basically the body is made of proteins? Proteins are the assembly of amino acids. And there are 23 amino acids, 8 essential amino acids. And, we've got, I don't know, 70 something thousand proteins. We also have, I think, 70 or 75,000 enzymes. And enzymes do the work, or a lot of the work, turning one thing into another. So, we all know about digestive enzymes. Where you eat protein, there is an enzyme that breaks it down eventually into amino acids. So are the genes the instructions, the blueprint, the architect's plan to make proteins, to make enzymes, and all the things that come from there?
Yeah, so some genes code for enzymes. So they basically are helping to speed up a reaction between 2 substances. So it's kind of converting A to B more quickly or not. So I kind of think of them almost as travelators. You kind of, that reaction, that chemical reaction is likely going to happen just due to different amounts of substance A or B being present. But the enzyme is the thing that pushes it along, gives it a shove, makes it happen more or less quickly. And it's the genes that are the facilitators of that. So they can, they are the controllers of all these reactions.
And we inherit variations of genes, which are called polymorphisms, many forms.
Exactly that. So we, even within the same gene, can have thousands of variances. So if a gene is 10,000 letters long, which isn't a particularly big gene, there are genes that are 50,000 letters long, but any one of those letters can be varied. It can be an alternative version. And so people have, just naturally, all of us have got lots and lots of variations in all of our genes, but we will never know about a lot of those because mostly they don't make any difference, at least not that we're aware of. But we do know about variances or polymorphisms that have impact. So the bigger the impact, the more likely we are to know about it, because someone will have been ill, or there will have been investigations into someone's health or physiology or mental health that have enabled people to unravel and understand these differences and their impacts. So we're interested in knowing about the variants that are well understood and that are common, because we can do something about them. And that really is the whole point as a clinician, to be able to action something differently because you've got some knowledge that you didn't have access to before.
And what is the evolutionary point in having different polymorphisms? Is there one, or is it just a matter of chance? How does this happen?
It's all about survival. So I think nature has hedged her bets and continues to do so. Some variances seem random, and they may just be random in the first place, but the more advantageous a variant is, so someone has a different version of a gene that speeds up a certain reaction, if that is advantageous to survival, then that gene version will be passed on. It's much more likely that that will be passed on and inherited. And so more and more descendants of that individual will hold that gene because it's helped them at least reach reproductive age. So that's the principle of inheritance, really. And sometimes there are multiple versions of the gene, and they are advantageous and disadvantageous in different ways and in different environments. So one is not necessarily good or bad, but it depends on the environment that that individual is living in.
It sounds a little bit like neurodivergence,
Yeah,
genodivergence.
exactly.
And actually, of course, it feeds right back into and makes one realize the absolute brilliance of Charles Darwin, who said that there are two forces driving evolution. One is natural selection, and the other, the conditions of existence.
Yeah.
And he said in every single thing that he wrote that it was the conditions of existence, in other words, the environment, that is the more important. And also that it is changes in the environment that then sort of natural selection then kicks in. There's some change in our environment or our food supply, whatever, and then certain polymorphisms, do better than others.
Yeah, and that there are loads and loads of examples of that are just really nice explanations of the whole thing, really. So, for example, lactose tolerance. The human default is that we cannot digest lactose, so the milk sugar, beyond weaning. So we lose the ability to do that. But almost half the human race can now digest lactose, because a version of the gene has evolved and persisted in order to help people survive, because it's been a live or die source of food. So, but our kind of default was not to be able to do that, and the ability to do so has evolved as we've moved away from coastal areas, started to depend on animal milk. That's how it all happened initially. But whether it's advantageous or not depends where you live, and it depends on your diet, and it depends on your culture and all those things. And some people would say it's not beneficial to drink milk, even if you can. Many different opinions on that.
And then, of course, apart from these polymorphisms, we also have causative genes. And once again, it was Linus Pauling who discovered the cause of sickle cell anemia as a genetic variant or a gene that one can inherit.
Yeah. So sometimes the genetic code will 100% result in a certain phenotype. So a phenotype is how something physically manifests itself. So sickle cell anemia is an example of that. If you have the genetic code that confers that, it will always happen that way. And there are other conditions that apply to that as well. But again, why would that condition of sickle cell anemia, why would it persist? There's got to be some advantage, right? And so the theory is that it's protective against malaria, because the cells can't be infected with the malarial mosquito passing on that disease. So because of their shape. And so there's an advantage to having that condition as well as a disadvantage. And if you're in a place where you're exposed to the potential of malaria, then actually you might have a better chance of surviving if you have sickle cell. So these things don't exist, at least not in any significant way or very common way, unless there is some kind of upside.
I mean, there's the other example, which is so fascinating. There's so few people really know, is that us humans are unable to synthesize vitamin C, which is synthesized in all plants from fructose, and all animals, virtually all animals. There's a few exceptions like the guinea pig, red-vented bulbul bird, most species of bats, all primates cannot make vitamin C. Pretty much everybody else can. And I think it's the loss of a codon, that there's one enzyme called GOLO.
Yeah.
And apparently we lack just a codon, sort of 3 letters or something. And there lies a big twist in fate. Is that right?
Yeah, that's true. And, you know, again, it's not necessarily a big change in a gene that makes a big difference. Sometimes it can be just the tiniest single change, or something like a codon is a relatively small change, but to have a massive effect. And it is really interesting. And obviously, in order for mammals to be able to survive with that variant, that polymorphism, they must have been in an environment where they could actually get vitamin C in food where it was available, because otherwise they wouldn't have been able to survive. And I think it's really interesting that fruit bats can't make vitamin C. So other bats can, but fruit bats can't. And they're called fruit bats, so they eat fruit. That's where they get their vitamin C.
Yeah, I mean, my story, and I'll just tell you it, and you can tell me if it's wrong, it's fine to do so, was, basically, because we probably don't know. I say maybe there's a zap of radiation, and some poor primate ancestor loses a little bit of codon, can't make vitamin C, which is made from glucose. And you use a lot of glucose to make a lot of vitamin C. I mean, animals produce many grams of vitamin C. So as long as they could eat it, it would have the effect of them having more energy.
Yeah.
And by having more energy, they became, well, probably top shagger, basically. And then we're all descended from that. And it may give humans energy, provided they live in a tropical jungle. The opposite if they live in a concrete jungle. And is that sort of probably the story?
Yeah, that does make sense, and it does ring a bell, actually. Absolutely, it's sparing them that energy that can be used for other purposes.
And I realized that Linus Pauling, he came across 3 things. One was we don't make vitamin C. 2 is animals who don't make vitamin C are much more prone to viral diseases, and cancer and heart disease. And it was just those simple logic steps that had him involved, and this is one of the most genius minds of all times, for 37 years. Just those very, so, very, very simple things. Now, let's move on a little bit to Alzheimer's and explore the difference and also what we can do between causative genes and maybe I would call them predictive genes, but the two causative genes for Alzheimer's, amyloid protein precursor and presenilin. So if both your parents have these, is it guaranteed that you will have those genes? And if one does, is it a 50-50 chance? And if you have it, does that mean you're going to develop Alzheimer's?
So it depends. Often with genetics, you can have, because you inherit 2 copies of a gene, one from each parent, some conditions only need one dodgy version of the gene to have a negative effect. Other conditions, you need to have both of those unhelpful versions of the gene. So you need to have inherited it from both of your parents. But let's assume, because I don't know for sure, but that you only need a partial inheritance. So we call it heterozygous. Hetero, it's a mix. It's one of each. You've got one good and one not so good version of the gene. If that's the case, then you may or may not inherit the dodgy gene from a parent that develops early onset Alzheimer's due to that genetic change. Whereas and then the risk of the inheritance gets higher if both parents had the negative version of the gene and if they developed the condition. So in that case, we know that the risk associated with that variant is extremely high. And you use the word causative, so it's almost inevitable that the inheritor of that version of the gene will develop early onset Alzheimer's.
And by early onset, we mean literally in 50s or 60s, possibly early in 70s, but people getting Alzheimer's in their 80s or 90s, that is not indicative of a causative gene.
No, these causative genes have their effects much earlier. It's almost like a much more concentrated impact, unfortunately. And it's different from the less determinate genes like ApoE, which I know we're going to talk about, because we don't yet know what we can do to really make a difference. And, again, that fits with the term causative. So we're starting to understand the mechanisms behind those variances. Like, it changes the shape of a protein, it changes the behavior of an enzyme and causes, we think, more amyloid build up, but we still don't know how to stop it. That's the real, shame of that picture. It's much harder to solve than some of the conditions that are associated with genetics that aren't so determinate. There is less ability currently to intervene.
Well, I'm glad you say that, because I've been given information from clinicians who have worked with people with causative genes for Alzheimer's, changing many of the things that we would do: homocysteine, B vitamins, omegas, less sugar, more brain activity, and so on. And their degeneration is much, much slower and better. So even having a causative gene is not necessarily, a guarantee of a certain path in relation to a certain disease. So let's talk about ApoE gene, which is probably the kind of most talked about gene of all. So first of all, in a very broad sense, what does ApoE do? What's it coding for? Is it an enzyme? Is it a protein? How does it work?
So there are a whole family of Apo genes. So there's Apo A, B, C, and E, and maybe there's a D in there in the middle too. But they all carry fats. So that is their job. They create a kind of, like, imagine it as a ball, which includes proteins, and that's what the APO part stands for, and fats, which we think of as cholesterol, and transport it around the body and the brain. And what these different APOs do is they kind of deliver it to different places and allow or not that cholesterol to be broken down into its different parts. And the problem with ApoE specifically is that it actually alters, and this is the unhelpful version of the gene ApoE4 that is associated with late onset Alzheimer's. It actually inhibits the breakdown of fats. So we call that lipolysis or lipolysis, and it prevents the fats from being broken down, and it also affects the ratio of the high density cholesterol and the low density cholesterol, so HDL and LDL. When ApoE4 is in the mix, we end up with a lower amount of HDL, more LDL, and also higher triglycerides in the blood. Now, we all know now it's not as simple as HDL good, LDL bad. There is much more to it than that. But basically, it's inhibiting the clearance, and it is associated with inflammation in our blood circulation and arteries and all those things. And we do know that inflammation is not a good thing when it comes to cognitive health, when it comes to cardiovascular health, and all those related things. So in a simple way, ApoE4, the negative version of the gene, is associated with higher inflammation and the consequences of that in our brains and in our arteries.
And in real numbers, to what extent does having ApoE4 increase a person's predicted risk of developing Alzheimer's?
It's quite high, actually. I mean, all these numbers, we could pick them apart and, critique them slightly differently. But the consensus seems to be that if you have the homozygous version of the genes, that means you've inherited the worst version, the 4 version from each parent.
So it's like having 2 balls.
Yeah,
Is that what you call a double allele?
exactly. So it's described as ApoE44, and that is associated with up to or about 50% higher risk, as in about 50% of people with 44 would develop Alzheimer's at some point, but it's late onset. So it's a slow burning thing. ApoE34, so the heterozygous, where you've got one risk version, one risk allele and one non-risk allele, about 30% of people are expected to develop late onset Alzheimer's, and it's also worse for women. So for men with a 3-4, they barely have any increased risk compared to a 3-3, which is the reference, the most common version of ApoE. But for women with 3-4, just one of these risk alleles, the risk is considerably higher.
Now let's just explore that, because one of the things that I did realizing this was to look at kind of fairly major studies, which were usually looking at some significant change like diet. And one comes to mind the year before last, 29,000 people, British Medical Journal, and looking at diet. And the essence was those with a healthy diet and lifestyle were about 11 times less likely to develop Alzheimer's. But in quite a lot of these big studies, when they're looking at two groups, good diet, bad diet, or placebo, or drug, or whatever, they're going to measure ApoE variants. And I believe it might have been something like 20% had ApoE. I'm not quite sure what the percentage of the population, what is the percentage of the population roughly who have ApoE4?
It's so the 4-4 isn't actually that high. It's about 4%, like 3.6 or something like that.
Yeah. So then what happened in these studies, they always had an outcome, better diet, less risk, and so on. And so, yes, I looked at most of the big studies I could find to say, was the outcome different for those with ApoE4 versus those not? And in almost all studies, there did not appear to be any difference once something had been changed. Better diet, more omegas, whatever it happened to be. How does that work?
So it's really interesting because, like all of these genes that aren't causative, as in it's not inevitable that the gene variant will lead to a specific outcome, there have been, as you say, studies, research done again and again that have shown, for example, in populations who have an ancestral diet that is their ancestral diet in terms of their culture and their environment, that even with higher risk APO alleles, they don't necessarily have higher risk of Alzheimer's. And one example of that, it's referred to as the Nigerian paradox, actually. So ApoE4 is more common in a lot of African populations. So taking Nigeria as an example. But historically, those populations, when they have eaten a diet that is their cultural diet that involves natural, unprocessed foods and fiber and all those things, their risk of developing Alzheimer's is barely increased at all. But that changes once those populations start to have access to processed foods and fast foods and, you name it, cheap outlet, etc.
Yeah, it's fascinating because, I mean, in a simplistic way, this may or may not be, obviously not the whole story, but it might be part of it. But it's a bit like, if you've got ApoE4, you're more likely to accumulate triglycerides, which are the blood fats we don't want. But of course, if you have a healthy diet, you're going to have less triglycerides. So, the diet massively, impacts that kind of thing. And I hadn't quite put the pieces together because I interviewed Lance Pauling literally a few months before he died in his mid-90s, when he was working on lipoprotein A as a driver of heart disease, which is now one of the hottest topics. And I do remember actually filming him, and he seemed to be going off track. He was telling these different pieces, and I felt, this man's total genius. Einstein, when when asked, "Are you a genius?" He said, "If you want a real genius, it's Linus Pauling." But I fortunately I didn't interrupt him, because he then said, "The reason I tell you these," and these pieces came together, and the essence of it was that his proposition was that when you're vitamin C deficient, you will produce more apoprotein. So that's what we're talking about here.
Right.
The carrier of fat, which inevitably attracts the fat, which is the lipid, becomes lipoprotein. And the reason you do this is because when you're vitamin C deficient, your arteries are going to start to degenerate. That's what scurvy is. And the production of lipoprotein will actually sort of thicken and fill the arteries, so prevent you dying from scurvy. So an evolutionary protection. So that also suggests, that kind of logic suggests that, maybe having a lot of vitamin C would also, to some extent, be another mitigating factor for ApoE4.
Yeah, that makes sense. I mean, all these things are connected, and that's why we have populations that, when you start to look at the genetics and the combinations of genetic variants that they have, it's kind of, well, they've got this, and that looks as if it could be risky and not very good for survival or health, but actually they've got this as well, and that interacts with the other and it offsets it in some way. So, natural selection and all of that has been pretty effective in coming up with that sort of solution, really. It's quite incredible.
And also, by being able to find out all these little variations, you're really getting an insight into how to personalize a person's environment. That is, primarily their nutrition, to kind of work with your pluses and minuses, so to speak. Now let's take a look at methylation, because methylation is such a fundamental process in the body. It does so much. I mean, tell us what methylation is, and how many genes are there related to the process of methylation, and how would they therefore impact upon health?
Wow. So methylation is a real buzzword at the moment, isn't it? It's just all over the place. It's a long word, and a lot of people think they know what it is, but they don't really. In a nutshell, methylation is the transfer of something called a methyl group, which is a carbon and 3 hydrogens, CH3. And this little compound basically gets removed from one substance and added to another. And the substance that it leaves is demethylated, and the substance that it gets attached to is methylated. So that's a kind of biochemistry of methylation. And something that is methylated, so for example, you can have methylated folate, methylated B12, behaves differently, it reacts differently biochemically than the unmethylated form. So it might be more or less reactive. And indeed, homocysteine, when homocysteine is methylated, it becomes methionine, and all the kind of nasty things that we associate with homocysteine, like inflammation and toxicity and linking back to Alzheimer's risk, become neutralized in homocysteine becoming methylated. So it's a bit of a magic thing, really. It's an incredibly important part of our biochemistry.
Doesn't it turn genes on or off, and
Yeah,
also protect or help to repair DNA and
yeah,
make neurotransmitters and several hormones and so
yeah.
on?
It's absolutely huge. I sometimes kind of think of it as magic fairy dust. It's just like, it's just doing so many different things. So yeah, it breaks down, deactivates neurotransmitters, things like histamine. It helps make choline in our bodies. That's hugely important. Of course, we can and should get choline from the diet, but
which is rich in eggs and rich in fish.
yeah,
And it's the choline or the phosphatidylcholine that attaches to omega-3 DHA to basically activate it within the membranes of all cells, but especially brain cells.
absolutely. So it's really, really huge. And if we're not getting good amounts of choline in our diet, then we'll try and make it, our body will make it, but it uses up a lot of our methyl resource in doing so. So it's very depleting. You know, there's only so much of this to go around. So if we're not having a good diet and doing what we can in that respect, then it kind of gets diluted, and the body is trying to, you know, prioritize and work out where to put the resources that it has.
So I either eat eggs or I have lecithin, rich in phosphatidylcholine, every day,
Yeah,
in the
yeah,
belief that I'll have more magic dust.
exactly. You will. That's it's really, really sparing of methyl groups. And we can get methyl groups from other sources as well. So from folate. So, you know, all energy, the solar energy is captured by plants, and we can eat those plants and benefit from the folate and other things that are in them, and use that to make methyl groups too. So lots of different sources of it. But of course, genes, again, you asked about how many genes are involved in methylation. So there are lots and lots of genes involved in methylation. The most infamous one, of course, is MTHFR. That's the one that everybody wants to know about, but seems to blame for everything. So MTHFR is the short code for a gene that basically creates methylfolate. So the folate that we get from the diet, it keeps it cycling around, and it keeps adding different carbons and hydrogens together to create this methyl version. And that really is the spark that stimulates methylation.
And that's what
So
we
it's,
want. We want methylfolate. We eat folate,
yeah,
or we may supplement folic acid. But that
yeah,
MTHFR enzyme, which of course is made by, so to speak, the gene,
yes,
is absolutely critical to make the stuff that works, the methylfolate.
yeah, yeah. So it keeps the folates kind of going around. So the folate is I can't say it now. The folate is converted, it's activated to methylfolate by that gene. Then the methyl goes and does one of its many jobs, and the folate loses it. It loses its methyl, and MTHFR keeps that cycle going to
Okay.
refresh it.
So you get many bangs for your buck if you've got good MTHFR enzyme activity. But what if you inherit one of the variations? I think the most famous is the C677TT, isn't it?
Yeah, that
Yeah.
is. So that can have up to 70%, 70, impact on that gene activity. And note the word up to, or words. Actually, we can almost totally compensate for that variance by supporting that gene with vitamin B2. So that's known as a cofactor. These nutrients and these genes don't work in isolation. There's lots and lots of other magic dust that can be added into the mix. It is like a recipe. to just help it along and optimize the activity. So, you know, I often hear people say, I've been diagnosed with MTHFR as if it's some sort of death sentence. And actually, all it means is you need to make sure that you've got good folate in your diet, good amounts of folate, eating it every day, some green leafy veg in there, and other B vitamins that complement that whole system. And guess what? When we eat food that has folate in it, a lot of the time it's got other B vitamins in it as well. So nature has packaged this food for us very, very nicely so that all the components that need each other are included.
And I know, yeah, you mentioned a very important point that enzymes have sort of cofactors, vitamins, minerals that are required for them to work. And this particular MTHFR enzyme has riboflavin, vitamin B2 as a cofactor. So you not only need the thing that you're trying to convert, in this case folate, but you also need the cofactor. Yeah, it was fascinating because Professor David Smith's brilliant research on homocysteine. Basically, we're turning methionine, it goes through homocysteine, and if we've got enough of the right vitamins, into S-adenosyl methionine, which actually does a lot of this methyl donor activity. And so, yes, what he did, so if your homocysteine goes up, you've got a log jam, you're lacking one or more of these nutrients. So homocysteine is a kind of functional marker. But again, in his study, of course, he'd measured those who did or don't have MTHFR 677, either one allele T or two. And again, I think that's quite common, isn't it? Or something like 20% of the population or something of that order?
Yeah, so nearly 50% of the population have at least one T allele. It's incredibly common. So that says to me, it can't be that detrimental. There's got to be more to it. Otherwise, there wouldn't be so many people walking around living their lives oblivious to the fact that they've got this difference. So there's always a trade-off, there's always a benefit. And I think so a potential benefit of that variance is when folate is scarce. So if we're not taking enough of it, then actually the folate that is there goes towards DNA repair and DNA synthesis. So it helps to keep renewing cells, but there isn't enough to go around and do everything. So the downside and the loser in that situation is the homocysteine and methionine cycle, and they're much more likely to experience high homocysteine and all the detrimental consequences of that may be longer term. So they wouldn't realize in the short term, it's like a smoldering, burning ember that is, beneath the surface, just causing damage and chronic inflammation. And it's only perhaps, in the 50s, 60s, 70s that they start to experience symptoms due to that.
And it's fascinating. It's really kind of important to know that, if you have MTHFR 677, maybe TT, it's sort of, the way I look at it is, in the darkness of an average poor diet and so on, it does confer extra risk. So lots of studies that show, more people with schizophrenia, more people with autism, more people with depression, more people with Alzheimer's, the risk is greater. But what's fascinating is, if you take something like David Smith's study, where they measured homocysteine, they gave vitamin B6, folic acid, not methylfolate, folic acid, and B12, and got an amazing outcome, in terms of reduced rate of brain shrinkage, better cognitive function, and so on. And again, I looked at the difference in outcome between those with or without MTHFR 677 TT, and there was none. So again, you have a classic example here where the nutrients are provided. And so that logjam is not so important because the pathway is still working. It just would work slower.
Yeah, it would work slower. And for some people, they might have a collection of alterations, a collection of variances that stack up and do make a difference. And I totally hear what you're saying. If everybody had a good diet and if everyone had high intake of these nutrients and it was a nutrient dense diet, then very few people would develop these conditions and these illnesses. But what motivates people to actually change their diet and take an interest in it and make the right decisions? Genetics can be helpful in that way. It can help people to focus on certain things if there is a logjam. So sometimes there is no particular logjam, but other times someone might present with high homocysteine and they think they're doing the right things. They think they've got a good diet. A lot of people think they eat well in today's world, but because of the proportion of bad stuff that is constantly pushed at us, it's often not good enough. So actually being able to know that if you personally do not take action and have more of this certain thing that you personally need, then you are much more likely to experience these consequences.
And are there some sort of methylation-related genes that are much more prevalent in, for example, Asians versus Europeans? Are there, how does that work? I mean,
yeah, so I think we looked at a gene called DHFR. And so what this gene does, it stands for dihydrofolate reductase. It basically converts dihydrofolate, so a form of folate, higher up the chain from an MTHFR. But when we eat folic acid, the synthetic form, so, for some people, folic acid might be okay and it might actually be beneficial, at least compared to them being deficient or insufficient in folate. But folic acid needs an extra step of conversion, and that DHFR gene codes for the enzyme that performs that role. And so people with that version of the gene, for them, folic acid may not be the best form of folate to use to support themselves.
so methylfolate would be good for them. And is that more prevalent in certain ethnic groups than others?
Yeah, I think Asian populations have got more prevalence of that DHFR variant. So, it's a little bit of folic acid probably wouldn't be harmful. As I said, it'd probably be more good than harm. But we do have situations where people are told to take really, really high dose folic acid. So, it might be related to fertility, for example. And actually, that, again, it might be okay in the short term, although there is research around fertility that says, what, actually people with this genetic variant do much better with methylfolate than folic acid. And actually for the long term, if they persisted in taking more than a little bit of folic acid, it could actually be detrimental because it can just jam up the whole chain of folate cycling.
Now, I listened to and thoroughly enjoyed your webinar at foodforthebrain.org, which
Thank
went
you.
into all these pathways. And, there are so many gene variations. But what I loved was, it was such fun in a way. It's like kind of personalized engineering that you can you almost get to see the biological character of an individual, and you, with your feet firmly planted in the world of Optimum Nutrition, can actually advise people about the important nutrients, the forms of those nutrients.
Exactly.
That would be optimum for them. So when someone does a test with you, like a sort of methyl gene test, just sort of describe in outline, the process and what they learn.
Yeah, brilliant. Yeah, I absolutely love all this stuff. It's as if everything in my life converged on genetics and nutrition and health. So you do a simple cheek swab test. So it's just like a cotton bud, you collect cheek cells. We can also use blood spot to do this, to collect DNA. Then the DNA is extracted at a lab. It's red. So we've basically set up a unique, it's called an assay, but it's a group of genes and variances that we know make the biggest difference to, say, methylation and are well understood. And so each of those variances, when we know that someone has them, there is also a course of action that can be taken to compensate for any detrimental effects. So we get the results back, the data, and we create this wonderful visual report that shows you where your hurdles are, where your blockages are, and most importantly, things like the cofactors that you can use to hop over that hurdle or cycle around it, to basically mitigate any of the risks associated with those potentially detrimental genes. So I love my job most of all because it's never really bad news. It's always about informing people about themselves and giving them something that empowers them to be healthier, take a preventative and personalized approach to their health. And a lot of people find it incredibly fascinating and motivating. So methylation is really at the heart of so many things. As Patrick, you said, neurotransmitters are another aspect, which is why I did that webinar for Food for the Brain. It's, methylation impacts how we make neurotransmitters, how sensitive we are to them, how good we are at regulating them and removing them. So that impacts and that connection applies in that context and lots of other contexts too.
And your website is LifecodeGX.com, is that correct?
That is the one, yes.
And very kindly, you've got a whole methylation panel, which you've very kindly given a hefty discount on for friends of Food for the Brain. And not only does the person get the discount, but you're matching that discount with a donation to help us continue our research, because we hope in the not too distant future that all the people who are doing the cognitive function test also can measure their homocysteine at Food for the Brain. What we'd like is to also start to include the ability to do some genetic testing.
Yeah. And that package, very, very importantly, includes support from a qualified nutritionist. So, this stuff is brilliant, but it's not necessarily simple. And we want to make sure people don't get overexcited about having an NPTH of arsnip and feeling panicky about it, because it's something that should feel really, really empowering. We think it's a great partnership with Food for the Brain. It's really, really good to complement genetics with the homocysteine tests and the other tests that you've got on offer there, because they will show you what your actual levels are, and the genetics will help to show you why. So it's just a fantastic partnership, and we're so happy to be supporting Food for the Brain.
And what is the link for that?
Yeah, so it's LifecodeGX.com, and then it's /FFB.
Fantastic. Emma, thank you so much. This really was a case of genes unplugged. I've learned a lot. I'm sure there are a lot of people out there who get very confused with all these words: alleles, polymorphisms, phenotype, genotype, heterogeneous, etc., etc. And you've made it all make tremendous sense. So thank you very much for being my guest on this podcast.
Thank you very much, Patrick. I've learned a lot too. I'm going to go and read more about Linus Pauling and continue to educate myself.
Thank you.
Thanks.